Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. In this presentation, we will present about vegetation analysis from Group 4. And before it, let me introduce our member group. Next. And then, member group of Group 4 is me, Mufti Ahmad Thoha, with study number 75, and then Ika Fani with study number 92, and then Randita Kusuma with study number 93. In this presentation, we have table of content. There is definition and purpose, concept, method, quantitative parameters, and community coefficient. Definition of vegetation analysis. Vegetation analysis is a way of studying the composition of type and form or structure of vegetation. The units of vegetation studied in the vegetation analysis is a plant community, which is a concrete association of all plant species that occupy a habitat. Purpose of width vegetation analysis. The purpose of vegetation analysis is to determine various types of widths from an agricultural area, study succession rate, determine width dominance, describe width composition, observe change in flora due to control, and determine appropriate width control measure from agricultural land. Studying the succession rate, knowing width dominance, knowing the description of width computation, observing change in flora due to control, and determining width control action. And then method of analysis with vegetation. There is 4 methods to analysis with vegetation. The methods are quadratic method, visual estimation method, line method, and point method. Quadratic method. The quadratic method is a vegetation analysis method based on an area of sample plot. Usually, the sample plot used are in the form of square, circle, and rectangle. The square referred to in this method is a measure of the area measured using square units such as meter quadratic and centimeter quadratic. And the second method is visual estimation method. Visual estimation is carried out by observing, seeing, and estimating with parameters at a certain point, which always remain in a limit sample plot. The calculated quantity is in the form of dominance, which is expressed as the percentage of spread.
Next, line method. Line method is carried out using an elongated sample plot that is placed on a vegetation community. The location of the area used is very wide. It can be done quickly and accurately. For example, for young plantation where wheat population are low in group with clear group boundaries. Point method. Point method is carried out like the extreme quadratic method, which is reduced to a point. This method is commonly used for low slope and creepers. Quantitative parameters. There are several quantitative parameters used to vegetation analysis. They are the percentage of distribution, density, frequency, and dominance. 1. Distribution. The percentage of distribution in addition to being expressed in present is often converted converted into 5 to 10 class of the abundance scale. Abundance scale class 1 really not common. It's here and there. General is a bit scattered. 5 very common plus yes. Weaver 1938. R is rare. A is infrequent. F is frequented. A is plentiful. VA is very plentiful. Austin 1956. Very rare, infrequent, abundant, very plentiful. Wirjaharja and Decker 1977 is plus 1, 2, and 3. Density show a type of plant in each sample plot in terms of coverage or basal area or biomass or volume.
The third quantitative parameter is frequencies. Frequencies of plant species are the number of sample plots, or in percent, contain that space from a number of sample plots made. In other words, frequency is the presence or absence of plant species in the sample plot. The frequency is influenced by the area of the sample plot, the distribution of plants, and the size of the plant species. The absolute frequency is number of sample tiles containing one type divided by number of all sample tiles taken. And the second is relative frequency, is absolute frequency value one type divided by total frequency value all type. Then dominance. Dominance is used to state how much area is overgrown by a type of plant or the ability of a plant species in terms of competing against other types. Dominance is expressed in terms of coverage or basal area or biomass or volume. The absolute dominance is dry weight or biomass type of a weight. Then second is relative dominance, is absolute dominance one type divided by total value of absolute dominance all type multiply by with 100,000. Important value or IV. Important value is the sum of relative density, relative frequency, and relative dominance or the relative number of parameters. Namely, the relative density or relative cover is biomass divided by the number or quantitative or parameters, which range between 0 and 300. The sixth value total dominance, NGD, is the number of significant values divided by the number of significant values. Important values are KR, or relative density, PR, or relative frequency, and DR, relative dominance. NGD is important value divided by 3. 7 species diversity. Species diversity can be present in the form of an index value using the Simpson index, namely here the formula of the ID. The information, the capital N is number of individual of all identified species, and the small n is number of individual of a species. And the last is community coefficient. To compare the vegetation communities or the type of vegetation from an area, use a formula. C is the community coefficient. 2W divided with A plus B multiply with 100%. C is community coefficient. W is the sum of the two lowest quantity for the species from each community. A is the sum of the entire quantity of the first community, and B is the sum of the entire quantity of the second community. This principle is also used to assess the variation of similarity of various community in an area. This coefficient can be obtained from quantity data or simple with a plus sign is yes or a strip sign is no, or from absolute or relative values for density, dominance, and frequency or SDR. In a herbicide experiment, it's necessary to calculate the for community coefficient, or C, between the sample plots to test whether there is a big difference. In the C value, for example, there is a 75% similarity, it is usually acceptable. Also, to compare a community before and after one control method, if there is a change in the flora.
Thank you very much for your time and attention. It's enough from us. Wassalamualaikum warahmatullahi wabarakatuh.